All right, translocations. Last time when we were talking about deletions and insertions, we were talking about when two chromosomes line up improperly in meiosis. And I showed you those illustrations where one chromosome can like loop back on itself and you can get some recombination happening. Or you could have two chromosomes and one of them's looping and the two chromosomes recombined. That gives you those inversions, deletions, and stuff. Those are all very rare. The cell is really good in meiosis about making sure that it's only pairing up homologous chromosomes and only recombining between chromosomes. But on these rare occasions, these weird things happen. Even more rare is that sometimes in meiosis, two different chromosomes will, for whatever reason, get lined up at the metaphase plate. Now, this is probably due to repetitive sequences. If there's a repetitive sequence in chromosome 4, for instance, and it meets a repetitive sequence that's kind of similar in chromosome 20, you can get some recombination. The cell will just make a mistake and think that those are actually homologous chromosomes just because they share some repetitive region. So what can happen very rarely then is chromosome 4 and chromosome 20 will line up in the metaphase plate, recombination will happen, and you get swapping of genetic material onto the other chromosome. So you will get what we call a derivative chromosome 4, that is, it's missing its big long arm here, and it's got now the short arm of chromosome 20 on the end of it. And then you have a derivative chromosome 20 with part of chromosome 4 arm on it. This will make it through meiosis, right? If it's recombined these chromosomes, at that point it's just going to separate the chromosomes in meiosis 1, and then it's going to separate the two sister chromatids in meiosis 2, and you will make fertile, functional gametes, eggs and sperm. You're probably not going to, in this case, if you've got a huge part of chromosome 4 and a huge part of chromosome 20, the resulting offspring are going to be lacking certain alleles, right? If you think about an organism that inherits this chromosome, it's missing a huge part of chromosome 4, right? And so the alleles that's going to be missing on that side, if that was an egg that was produced, an egg that has that chromosome, the sperm that fertilizes it is going to contribute most of chromosome 4's information, right? Because it's lacking a bunch of alleles on chromosome 4. So you can get offspring in the next generation that will actually be resulting from this. That individual is probably going to have problems making their own gametes, though. Because when they go to try and align their chromosome 4s, they're going to have a big long chromosome 4 that's the normal one that came from the dad, but then the mother's chromosome 4 is going to be really abnormal. So you might actually make it as an individual and be a fully formed functional individual offspring with this chromosomal abnormality, but probably you're going to be sterile because you can't line up your chromosomes properly. So things will stall in meiosis and you won't be able to make eggs and sperm. There are some places where it looks like chromosomes have recombined, and if it's just a little bit, if you just took, instead of the whole chromosome 4 arm, if you just got a little chunk of it put on chromosome 20, that would probably still be a viable offspring. But huge ones like this example are probably going to be lethal in the second generation, right? Okay, I want to talk about frame shifts now. We've talked about all of most of the ways that chromosomes can be mutated, right? Single polymorphisms, just single nucleotide changes, insertions, deletions, translocations. I want to talk about a frame shift, and this is a certain type of effect that a mutation will have in coding region, okay? So we're not talking about the intronic regions or intergenic space between genes. I'm talking about the actual exons, the actual coding region. If you have mutations that induce a frame shift, they're going to be very, very toxic. Typically, it's going to be insertions or deletions that are going to do frame shifts. But you could have frame shifts for another reason, which I'll get to in a second. Okay, so a frame shift, this is the simplest frame shift example here. Here is a normal DNA template, so one strand of the double-stranded genome. And this is the codons that are being used, right? This is TAC, ACC, GAG, okay? The codons, this is called the reading frame, is which series of 3 nucleotides do you use as a codon. The way the reading frame is set up is where is the start codon, right? The methionine TAG, ATG, sorry, ATG. TAG is a stop codon. ATG, right? That starts the first methionine, and that defines the first the first amino acid that's put in there. If you have a shift of that, right? That wherever you find the ATG as the start codon, that defines that's the first codon, and then the next 3 define the next amino acid. The next 3 define the next amino acid, right? So if the ATG was here, that has determined what frame I was in. Okay. Now you can shift frames. You can have multiple frames on the chromosome, right? I could read in this frame, but if the ATG was shifted by 1 base, I could read in a separate frame, right? I could shift over 1 base and read in a next frame. You could shift over 1 more frame, read the third base would then be the first one. But you can only do 3 frames, right? You can do the first one, the second one, the third one, but then a 4th frame would just be returning to the first frame. You understand? So this might be clear when we talk about a frame shift. But, well, actually, let's go over it. This would be the first frame, right? TAC, right? If we shifted over 1 frame, then my frame would be ACA, right? That would be my codon. That would be the second frame. The third frame would be if this C was the first one, CAC, right? The 4th frame, though, I've just returned to the original frame. Okay? So there's 3 reading frames possible on 1 strand. You can also read DNA on the other strand, right? I'm just showing you one of the strands, but DNA is double-stranded. There's a reverse strand going on, so there's 3 possible reading frames on the reverse strand, okay? So in a frame shift, what happens is a mutation causes you to shift to an alternative reading frame, okay? So here is my insertion. This little T has gotten inserted here between the C and the G. Okay, C and G were here, now I've inserted a T. Well, these first 2 codons are the same, TAC, TAC, ACC, ACC. But since I've inserted that T, I have shifted the frame. So now instead of reading GAG, now I'm reading TGA. And I'm going to start putting completely abnormal amino acids in this protein now. Usually, a frame shift, if you just shift the frame like this, you're probably going to start reading, number 1, you're going to put in inappropriate amino acids for a little bit, but probably you're going to have induced a new stop. A new stop codon will show up if you shifted the reading frame. So if you actually just look at DNA, if you just take a sequence of DNA and look at it, you can usually identify the coding region because it'll be a big, long place where all of the reading frame doesn't code for any stop codons. And you can say, oh, that's probably the exon in the genome. If there's a bunch of stop codons, then you're like, well, that's probably not a coding region, right? So this would be detrimental for the protein in 2 ways. Number 1, you're going to have, from the point of the frame shift, you're going to be putting inappropriate amino acids, and you're probably going to come across a stop codon pretty quick, and you're going to make a truncated protein. Okay. So anything after the insertion in the mRNA is now going to code for the wrong amino acid. If you insert 1 nucleotide or 2 nucleotides, you're going to have shifted the frame, right? If you insert 1 nucleotide, I've just shifted from the first reading frame to the second. If I insert 2 nucleotides, I've shifted from the first reading frame to the third reading frame. If you insert 3 nucleotides, you don't change the reading frame. All you've done is an insertion of a single amino acid, right? So if you have insertions that are in multiples of 3 nucleotides, if it's 3 nucleotides or 6 nucleotides or 9 nucleotides, then you won't change the reading frame. All you will have done is just put in a couple of extra amino acids. So frame shifts are always just, a single nucleotide or even numbers of nucleotides are going to induce a frame shift. If you have multiples of 3, you won't have changed the frame. that make sense? Okay. Sorry. All right, so frame shifts are very detrimental. Although there's weird things, especially in bacterial genomes. Bacterial genomes will have an open reading frame, a region that's going to code for a gene. And you might actually be able to code for 2 proteins off of the same sequence of DNA. One functional protein might be read in one frame, but there might be an alternative start site, an alternative ATG that sits in a different frame. And then you could actually make a completely different second protein off of the same DNA region because you're just reading it in a different frame. If that different frame doesn't induce a stop codon, it has the potential for coding for a second protein. And so in theory, you could actually, if you designed it properly, you could make 6 different proteins from a single piece of DNA. Because you could read in 3 frames one way, and you could read in 3 frames the other way. If you just made sure you didn't have any stop codons, you could actually make 6 possible protein sequences. And bacteria actually do this. They overlap their genes in their genome. They have such a small genome, and they got to get all the genes in there, they will actually overlap. And you can actually code for multiple things in the same location, which is bizarre then. I mean, it sets up a problem of which reading frame do you need to start with, right? How do you determine which protein is appropriate to make at this time and sit down RNA polymerase on the right start codon? And it's also kind of tricky code, right? If you're gonna, you have to make sure that you stop the proteins appropriately, but make sure that in the other reading frames you don't have a stop codon. So it's a pretty clever way to do that. Okay. In the context, if it's a defect, though, if it's a mutation, cells have very clever ways of repairing the DNA. Okay. And there's 2 main ways to repair DNA damage. Number 1 is reverse the damage by a direct reversal mechanism. That is, if there's a way to recognize if a wrong nucleotide got put in, or if an insertion got made, the cell can sometimes tell, based on the homologous chromosome, what the original sequence was. Okay? So this is where it's handy to be a diploid organism. If you're a diploid organism, you've got 2 copies of the genome. If one of your chromosomes mutates by getting, say, for instance, an insertion, well, you do have the other chromosome. And so some DNA repair mechanisms will actually pull the homologous chromosomes together, read one chromosome, and change the actual sequence on the other chromosome and reverse the damage. Right, so it will recognize this is an insertion point that my other chromosome doesn't have, and will actually pull it out. Right? So that's the 2 types of damage. You can either directly reverse the damage. This is like DNA polymerase with its exonuclease activity. Right? It's going along, it puts the wrong base in. Well, it just backs up, removes the base, and puts the correct one in. Removing the damage completely is the second thing, where if you can't just back up, right, if this is a bigger defect, then you actually go in, you remove the incorrect sequence, and you fill in with proper sequence, right? So this would be removing the damage and starting over. We call this excision repair, right? Because I'm actually going to have to cut out those bases and refill them in. I've been reading interesting things about this phenomenon called biased gene conversion. This is a weird phenomenon where, and I'm not prepared to talk about this, so it's going to be off the cuff here. But there's a tendency for chromosomes to correct mutations in certain ways. So this is kind of a problem for thinking about population genetics. If you have a new allele show up by mutation, new alleles will sometimes completely take over the population very, very quickly, quicker than it would just take for that to be a beneficial allele. Okay, so if you've got a new induced mutation, right? I was a homozygote at the site, and then I have a mutant allele, right? if that new mutant allele is going to take over the population, we're going to have to wait multiple generations, right, for it to segregate in meiosis, 2 new fertilized eggs come about. That new allele has to continue to just take over the population. Every time that mutant gets combined with another wild-type version, I've got a heterozygote, right? There's this thing called BIOS gene conversion, where certain alleles will actually induce the cell to change the other chromosome to be that same allele. So instead of being a heterozygote, you get a new allele, but you've got a wild-type allele, you're a heterozygote. When that individual makes its gametes, it will actually change its 2 alleles to match, and it will act as if it's a homozygous for the new allele. And so it's called biased gene conversion, because it's biased to convert the other chromosome to this chromosome's allele. So you go from being a heterozygote, but when you actually make gametes, you make gametes all of one allele, and you completely ignore the other allele that you have. This is called biased gene conversion. And it's bizarre, too, because how does the cell know which allele is the better one to give? And then it just throws off all of the idea of population genetics, because population genetics assumes that, by traditional Mendelian, if you're a heterozygote, then you've got a 50-50 chance of giving either allele, right? But biased gene conversion says, no, you're a heterozygote, but when you make your gametes, you bias them to one of the alleles. So you act as if you're a homozygous. So this is an interesting phenomenon that gets to kind of interesting things about how a cell works and how your organism works. Phenotypes are not as dictated upon your genes and upon your alleles as we kind of like to think they are. Because we think, well, your alleles are a heterozygote, right? And so we predict by Mendelian that you're going to act like a heterozygote. But the cell is deciding which of those alleles to use, right? So there's some other information going on. Your genotype does not always dictate what your phenotype is, right? Your cell might actually change your genotype to fit whatever phenotype it wants to express. It sets up this difficulty with genetics of kind of a chicken or an egg problem, right? Are your phenotypes a result of your genotypes, or are your genotypes the result of the phenotype your cell wants to do, right? Does the DNA dictate what you look like, or does the cell decide it wants to look like a certain way, and so it changes its genome so as to look that way? This is difficult to, it's difficult to see and test for, but then it's also really difficult to actually make predictions then about what organisms are going to look like and how they're going to change over time. That's a total tangent, but we'll talk about the mechanism of repair. So this idea, though, that the cell can repair, can bias its repair, and we've talked about editing before, too, that even if you have a mutation in your genome and you make an inappropriate mRNA, the ability for the cell to edit that mRNA will actually allow the cell to say, well, I'm a mutant, but when I express my mRNA, I change it so that it's still a functional protein. So you can have mutant alleles that get carried over multiple generations, but you never see the phenotype of that mutant because the cell is changing the mRNA and masking that mutation, right? So, Rachel.
I'm just curious what, according to natural selection, felt about that. Do they think that was extra support to their, or?
No, this is a real difficulty for natural selection, right? Because if the cell is changing its genome, then how can natural selection act on selecting alleles, right? If the alleles are hidden from the environment, right? If the organism is expressing its phenotype despite the genetic changes, then how can natural selection be selecting for certain alleles, right? Natural selection can only select for phenotypes, right? If I have a deficiency in metabolism or something, then I'm not going to be able to eat certain things, right? And so if certain things are not available to me, then my phenotype of not being able to metabolize things, I might be selected against, right? If I have limited food choices, if certain things poison me and they don't poison you, well, then I've got lower fitness level, and I'll be selected against. But if you can't correlate my phenotype to the genes, because it's the genes that get passed on for inheritance, right? So if the phenotype doesn't correspond to the genotype, then where is evolution at then, right? So this is a real difficulty for natural selection, and there's actually a lot of papers being written right now saying natural selection is probably not the whole story. We'll return to this when we talk about development too, because there's a lot of things that go on in development where changes in genes are going to have multiple functions or functions at different times, and so again, it's hard to pinpoint a certain nucleotide sequence to a certain phenotype. Just really quick, if you've got a gene that dictates, I don't know, the development of your brain, but that same gene also dictates your kidney's function, how does natural selection work on that, right? If you're selected against a certain allele because it decreases your kidney function, but you're selected for because it gives you higher functioning brain capacity, if a phenotype can't be corresponded directly to a single genotype, then natural selection really kind of becomes deficient in explaining how things change, right? You would say, if it's doing two functions, it's probably not going to change at all, right? Because increase in fitness of one area is going to decrease fitness in the other, right? So how could you select for that for a difference in that gene if selecting for a better kidney gives you a worse brain, or selecting for a better brain gives you a worse kidney? If two natural selection things are working against each other, then that wouldn't help in terms of evolving, changing over time, right? It more describes how things would stay the same rather than how they would change. But we'll get into that in development. All right. Direct reversal. We talked about this. Exonuclease activity, right? The polymerase just removing its inappropriate base. If your mutation was adding a methyl or ethyl group, like we talked about last time, the ENU molecule added that ethyl group to the thymine bases. There are some repair mechanisms that go in and recognize ethyl groups and remove them off of the nucleotides. So you can get repair enzymes that remove any of these foreign groups. And if you can't remove the foreign group, well, you might be able to just go in and remove the single base, right? It recognizes, I don't know how to remove that ethyl group off of the thymine, but at least it can recognize that's an inappropriate thymine, and so it'll just cut it out, put the new proper nucleotide in, right? So this would be an exonuclease or an endonuclease, goes in, clips out the inappropriate base, and DNA polymerase comes in and repairs it. You can do direct reversal if you've got the complementary strand there, right? If the mutation has just happened on one strand and you've got the appropriate non-mutated strand, repair enzymes can just come in, remove nucleotides. It's got the complement there. It can just put in the appropriate nucleotide. If both strands are mutated, then you probably can't do direct reversal. You're going to have to clip out both strands. If you clip out both strands, you don't know what the complementary strand was anymore, and so you're going to have a hard time repairing. Now, like I said, sometimes you can bring in the homologous chromosome, and that can help you repair. But usually direct reversal is only possible if you've got the complement there for you. All right. Does anybody want to talk about DNA repair? Yeah.
Sorry, how does the cell recognize these before it's too late, like before it's starting protein?
It depends on what kind of cell you are. Bacteria are generally pretty bad at detecting this, and they just let them go. And it's kind of, bacteria's way of life, right? Bacteria just make a lot of offspring. You duplicate your genome, you bud off a new daughter cell, and she's on her own to kind of just compete with whatever allele she may have gotten, whatever mutations may have been induced, right? If you're a multicellular animal, then you still have your cells around, and they could then be potentially harmful to you. So multicellular animals are going to have way more repair enzymes and checkpoints in their cell cycle. A bacteria doesn't really have many checkpoints in its cell cycle. Like if it duplicates its genome, it's probably going to divide into two daughter cells. If a multicellular animal goes through S phase and duplicates its genome, there's a checkpoint at the end of S phase, and there's a checkpoint at the end of GAP2, and there's all these checkpoints where these different enzymes come in and they proofread the genome, make sure Do I have all the copies of the chromosomes that I'm supposed to have? Are they all the same size and are they lining up? Do I detect any mutations? A lot of cells in multicellular animals will induce mutations, and if the mutation is too extreme and the cell recognizes I can't repair this by any known cell mechanisms, it will induce apoptosis, and the cell will just die, right? Rather than risk having a cell in your body that's now replicating that might be mutant and give you some abnormality. So if you can't do one of these repair mechanisms, often cells just commit suicide and die. Yeah.
So is cancer considered like can that be
Say it again.
So you say cells because they recognize that a gene that's important, it doesn't function properly, that they just go through apoptosis. So
Yeah.
is that, like, can that be characteristic of cancer?
Yeah, usually cancer, you've got a mutation, and often the mutations are in proteins that are regulating the cell cycle, right? So if you've got a mutation in one of these genes that recognizes DNA damage, and that one's no longer functioning, then you can have more extensive DNA damage. That protein is not performing its checkpoint function, right? It used to be the bouncer to make sure that cells only completed the cell cycle if they were appropriate, right? But if that protein, that bouncer protein, is mutated, well, then it's just going to keep going through the cell cycle whether there's mutations or not, right? So a lot of cancers are defects in these genes that regulate the cell cycle and make sure that these checkpoints, these criteria for cell health have been met, right? So these are tumor suppressor genes is usually what they're called, yeah. So a tumor suppressor protein is a protein that makes sure that mutated cells don't continue to divide. But if that's mutated itself, just kind of an unfortunate thing to have a mutation in your mutation repressing gene. That's bad news, right? Because then that cell is going to inappropriately divide. Yeah. Other questions about DNA repair? All right, let's talk about bacteria. Bacterial genetics, in a way, is kind of an oxymoron. Because when we think of genetics, you usually think of passing on genes to the next generation through sexual reproduction, right? All this time, we've been talking about genetics, and we've been talking about how it's so closely linked to what's happening in meiosis, right? The making of eggs and sperm, aligning homologous chromosomes, recombining them, creating gametes, doing sexual reproduction, right? That's how traits get passed on. Well, in bacteria, they don't reproduce asexually, right? A bacteria just duplicates its genome, sets up a spindle, separates its chromosomes, and then just divides in two, and there's no contribution from another cell, right? So they don't make germ cells. There's no fertilization. Most bacteria are haploid. Well, I should say they're monoploid. They only have one copy of the chromosome. So when they reproduce, they're just reproducing their own genome. Now, very rarely though, and only in certain instances, bacteria can actually though exchange some genetic information. Now, it's not in the form of two cells having copies of the chromosomes merging together, but they do have some sharing of genetic information. And I'm going to talk through these three types of transferring genetic material. Number one is conjugation. Conjugation is two bacteria agreeing to exchange some genetic information. We'll talk about how that works. Transformation can either happen naturally or artificially. It's a bacteria who's usually stressed. You stress the bacteria, it thinks it's on the verge of death, and it induces this program where it just starts bringing in things from the environment, just kind of as a last-ditch effort. Maybe there's something in the environment that will help me get over this condition. And so what they can do is actually bring in DNA from the surrounding environment. So that's transformation. Some bacteria will actually do this on their own. We'll just pull things in. Or you can actually induce them, force them to bring in DNA from the environment. So we're actually going to transform bacteria in 2 weeks. So the DNA that you've amplified is from Drosophila. We're going to put it in a bacterial chromosome, and then you're going to force bacteria to actually take in that recombined chromosome that you made. The third is transduction, and this is when viruses actually inject their DNA into bacteria. So there can be exchange of information through a viral intermediate. So I'm going to talk a little bit about bacteria, bacterial life cycle, and how we can actually look at bacterial phenotypes. Most of what we know about bacterial genetics has been done in E. coli. E. coli is a really great model organism, and it gets a bad rap because most people think of E. coli only when we're talking about the malicious disease-causing form of E. coli. There's actually very few strains of E. coli that actually cause really bad digestive stuff. Your gut is just filled with hundreds of thousands of E. coli right now, and they're all doing really good things to help you digest your food. It's only these bad disease-causing strains are the ones that you don't want. So E. coli is really common, very easy to culture. It's called the workhorse of molecular biology because just every lab uses it for just really common stuff now. So you can almost not be in any biology field and not use E. coli. You used to just be able to be a, ecologist or something, and you just go out in the field and just look at samples of organisms in the field. Now almost everybody goes out in the field, but then brings back DNA and manipulates it. And if you're going to manipulate DNA, you probably are going to go through E. coli at some point and use it as your workhorse. It's got a really small genome, only 4.6 megabases. So just right around 4.5 million base pairs, which is actually kind of a, comprehensible number. You can kind of wrap your head around 4 million. When you get to 3 billion base pairs in the human genome, that just starts staggering your ability to think about it, right? It's really easy. You can grow it in liquid. You can grow it on agar plates, as it's growing here. And it takes a pretty simple minimal media, right? It can do most of its own biochemistry, so you don't have to provide it a lot of nutrients, just basic nutrients, and it'll grow. We've sequenced this genome. In fact, I believe it was the first organism to have its genome sequenced. So way before the human genome, in the 2001 is when the human genome came out. 1997 is when we came out with the bacterial E. coli genome. Bacteria don't have very flashy phenotypes. We've been talking about like coat color and interesting things in terms of like plant phenotypes of flower color or the seed shape or things like that. You basically have a bacteria or you don't have a bacteria, and it's pretty hard to see a phenotype if it's just a bacteria growing on a plate, right? So here you got no plate, here you got bacteria growing on a plate. That's pretty much it in terms of the phenotypes, right? So all the phenotypes that we talk about usually in bacteria are going to be whether or not the bacteria can survive in the media that we give them. What are the nutrient requirements for the bacteria to live is mostly what the bacterial phenotypes are. Can the bacteria live if there's no methionine amino acids in the solution that's growing in? The other is, can it resist antibiotics? So those are the two main bacterial phenotypes. Can it survive without a nutrient, or is it dependent upon a nutrient? Or can it survive if there's an antibiotic that's going to kill it? Okay. So most of these are genes in synthetic processes in biochemistry in the bacteria. Right? So a MET plus bacterial strain would be a bacteria that is capable of metabolizing methionine. And so you could grow that bacteria in a plate, in its nutrient agar, just its nutrient culture, liquid culture, without methionine. So a MET plus strain can make methionine. You don't have to put it in the solution. Right? A MET minus bacteria would be one that's incapable of making methionine. So if you're going to grow that bacteria, you have to put it in a methionine-containing media, right? So when we look at bacterial phenotypes, we're going to be usually talking about what media do you put them on. A minimal media is just what does it take for a fully functional wild-type normal bacteria to grow? Not very much. It's got to have, I'm being attacked. It's going to have sugars, right? It's going to have certain nitrogen sources. It's going to have essential minerals. But mostly bacteria can just take these pretty basic forms of sugars, of molecules, and they can make all of their proteins, all of their DNA, all of their RNA. There's not a lot of vitamins that are required by bacteria, right? They can make most of what they need. So when we're talking about phenotypes, we're going to be saying, well, what do we have to put in the media? We have to supply a MET- strain with methionine in the media. There are certain vitamins that it needs. Biotin is one of them. So a biotin plus is a bacteria that can make the vitamin biotin. A bio- is a strain that you have to put biotin in the solution, in the media. Also, what sugar sources they can use is going to be a phenotype. Bacteria have the ability to metabolize several sugars. They can metabolize glucose, fructose, sucrose, galactose, and they will preferentially metabolize certain sugars. But there's certain strains that are defective in this. So a Gal plus strain is one that can use galactose as its sugar source. So it will preferentially use glucose, but if there's no glucose in the solution, it can use galactose. A Gal- strain would be one that can't use galactose. You either give it glucose or not. If you don't give it glucose, it's going to die. It's not going to be able to metabolize any galactose in the solution. Okay. And then antibiotic resistance, right? BLA plus. This is beta-lactamase. When a bacteria is, there's several ways that a bacteria can be resistant to antibiotics. Depends on how the antibiotic is functioning. In ampicillin, ampicillin will kill a normal bacteria because the ampicillin is brought into the cell and then it messes with the function of the bacteria. If the bacteria ignore the ampicillin and don't bring it inside, then it becomes resistant to that bacteria. So ampicillin resistance is actually, in a sense, a defect of a normal functioning bacteria. A normal functioning bacteria has these channels in its membrane and brings things into the cell, and it will bring ampicillin in the cell, which kills it, right? So it's non-discriminatory, but that channel is important. It brings in certain, and I should know more about this than I do, but it's a normal thing that wild-type bacteria use, and it's a part of their normal metabolism. A strain that's resistant to ampicillin make a non-functional channel, and they don't bring the ampicillin in. So in the presence of ampicillin, it's a benefit because you don't die, right? But you actually don't metabolize things as well, as efficiently. So if there's no ampicillin around, a wild-type bacterial strain will outcompete the antibiotic resistance. If there is antibiotics around, the antibiotic resistance will compete out the wild type. So it's kind of a balance between what's the beneficial trait to have, right? If you're just normal functioning in the wild, it's better to have that good channel and be able to bring in the nutrients that you want. If you're in an individual who's taking antibiotics, it's more beneficial to not do your metabolism as well, but at least survive, right? So there's different ones, but ampicillin or antibiotic resistance is another phenotype we're going to look at. So basically, though, we're just going to look at plates. Can a certain strain survive on a minimal media, or does that strain that I'm growing have to have a plate that's got certain nutrients in it? All right, conjugation. This is the first way that bacteria actually exchange genetic information. Conjugation is transfer of genetic material from one bacteria to another the transfer is unidirectional. Okay, so it's not a real exchange, it's just a gift from one to another, all right? There's no reciprocal exchange here. And cell-cell contact must be made. I'm not going to go through the experiment that they did to determine this originally, but we know now that one of the bacteria that's actually going to give the genetic information to the other, makes a little apparatus called a pilus. And it's this long, basic long tube or long straw that connects the cytoplasm of the giving guy to the receiving bacteria. So it makes this long tube, and then the genetic material is passed through that tube into the receiving, and then the tube is broken and the two part ways. Right? So it's just a single event. It's a genetic transfer from one to the other. The underlying genetic reason for this is that the giving bacteria contains a plasmid called an F factor. So here is the giving bacteria. And it contains, in addition to its genome, so in this illustration in blue is the bacterial genome. It's normal functioning genome. It's got all the stuff that it needs to actually exist as a bacteria. In addition to that, it's got a little accessory chromosome called a plasmid. This is the F factor plasmid. If it contains the proteins that produce this pili, this little tube that it's going to make to meet up with the other bacteria. So on that F factor plasmid, there's an origin of replication, right? There's a site so that that plasmid can actually get made again when this cell divides. So if you're this cell and you've got the F factor, when you divide, just during normal cell division of a bacteria, you're going to replicate your entire genome, and you're going to replicate the plasmids, and then the two daughter cells that result from that asexual reproduction are both going to contain the F factor. Right? So it's inherited through daughter cells through asexual. But it also, on it, has several genes that produce this pilus, this long tube. Okay? And when those genes get turned on and they actually start making this F pilus, and we're just illustrating it by this little channel here, they actually reach out and will only make contact with bacteria that don't have the F pilus. Right? They recognize each other, and if it doesn't contain the F factor, this guy, then this one that does contain the F factor will make a pilus and will send it a copy, one of the copies, and there's many copies of each of these plasmids. So it's not like it's just giving up its single one, it's just giving one of the many that it has. That F plasmid gets replicated, and as it gets replicated, it injects it into the what we call the F minus, the one that doesn't have the F factor. And now this one has converted from an F minus and now exists as an F plus bacteria. That guy now has the genes to make the pilus. It can go on and do conjugation with some other F minus. Right? So this is normal conjugation with just kind of a bare bones F plasmid. So the only information this is really exchanging here is just the ability to exchange information again. Questions about how this works? The interesting part comes in these strains that are called HFR strains. HFR stands for high frequency of recombination. And what happens in these is this F factor that exists in this bacterial cell actually recombines with the genome. And it inserts itself into the genome. Now, the reason this is interesting is because if this guy has an F plasmid inserted into its genome, basically the bacteria think that the entire genome is the F factor. So when this high frequency, this is a recombinant individual, right? It had an F factor, but that F factor recombined into its genome. Well, when the genes get turned on to make the pilus, and it actually undergoes conjugation, it's going to replicate, it's going to start replicating the F factor like it would normally have done, But it replicates not only the F factor, but part of its own genome. And so it passes not only the F factor, but genes on its actual genome to the bacteria that it's conjugating with. This is where you actually get some new genetic information being exchanged that kind of makes things interesting, right? Because now genes that were sitting on this genome are in the second bacterial genome, and now I actually have a in a way, pseudo diploid organism here, right? I've got all of the genes that were on that original genome, but now I've got another copy of genes that came from another individual. So for the most part, bacteria act as if they only have one genome, right? They've got only one allele of every gene. But in these HFR strains, when they conjugate, you can actually get the resulting guy to act like a diploid organism, right? For whatever, however much of the genome it inherited, and it's not going to get all the way through the genome. It's just going to give some genes that are sitting next to it on the genome, because it actually takes more time to replicate the entire genome. It's not all they're going to get over. So you're going to have the F factor and then just some things that flank it, right? So for some genes, it will be a diploid organism. For other genes, it's going to still remain haploid or monoploid, I guess. Now we can actually talk about bacteria inheriting traits and having I don't know, phenotypic differences between them, right? Because if you just have this guy in a culture dish, then basically every offspring, every daughter cell is going to have the exact identical phenotype, right? Because every daughter cell is just inheriting the same genome. But now I've got chance for recombination here between the two genomes, and now daughter cells from this cell might actually show different phenotypes depending on which alleles they inherit from during replication. Yeah, one question, then we'll have to go.
So the bacterial genome can recombine with their plasmids?
In these weird HFR strains, yeah. This is not typical. A normal wild-type bacteria doesn't usually recombine with its plasmids. But in these weird strains, and I'm not sure what the background in these strains is that makes them do this, but it's just these HFR strains that do it. So most of this is like stuff we've been doing in the lab, not really what's going on so much in the wild.
Biola University offers a variety of biblically centered degree programs, ranging from business to ministry to the arts and sciences. Visit biola.edu to find out how Biola could make a difference in your life.